to host you here at the BioData Talks this afternoon. And we have Simon Cox and João Machado as speakers. And they will talk about their works on the marine resources community that they coordinate in BioData. It is noteworthy that CCMAR is also hosting the Portuguese node of MBRC. So we have here today two research infrastructures that collaborate in this endeavor of making available marine resources for research. Thank you very much, Simon and João, for accepting this invitation. And without further belongings, I will pass the floor to Simon.
Thanks, Anna. So again, welcome everybody to this afternoon's talk. It's going to be pretty short, I think. I have about 10 slides, and then my colleague João is going to present another 10 slides. So I'm going to do a brief overview of the community and what we've been doing and some of the projects we have. And then João's going to talk specifically about a project that we started quite recently to build a database of seagrass phenotypic data, which is quite interesting. So we'll get to that later. So let's say something first off about the status of the community. If you don't know, I mean, BioData.pt is the national node of ELIXIR, and ELIXIR is the EU F3 research infrastructure for bioinformatics in Europe. So that's the status of the project. And within this project, in 2013, we set up a marine genomics community, which was basically set up to mirror the ELIXIR marine metagenomics community. So we have like a national level community, which then feeds into the more general European one. But the problem with that seems to be that it was a bit too exclusive, you know, tying the community to basically to the marine domain and a specific, you know, methodology excluded too many people. And beginning next year, they're actually going to change the community to be the ELIXIR Microbiome Community, which I think is a very good idea. It's going to be more inclusive, and I think more people will get involved. And BioData.pt is also going to mirror that as well. So there will be a microbiome community for BioData.pt, and I think that's going to be more inclusive on a national level as well. So this community we're talking about today is the Digital Marine Resources Community, which follows on partly from this marine metagenomics community, basically just dealing with the marine domain and digital resources that we have in Portugal. So the objectives of the community, well, basically, we want to provide a forum for researchers working with digital data in the marine environment. I mean, that's our basic goal, and to also provide digital services, training, you know, computational resources, and other virtual workspaces within the digital domain for marine biologists in Portugal, and also to provide an ongoing assessment and re-evaluation of the community needs. I mean, it is obviously changing, and those needs will change as technologies change, and as as the community changes at the European level, initiatives feed down into the national level as well. So one of the things that we've spent most time on is actually installing these computational equipment that we have here at CCMAR, which is actually funded partly by BioData.pt and partly by EMBRC. So we're also the Portuguese node for EMBRC, which is the European Marine Biological Research Centre. And we've bought and installed a system which is virtualized on a Microsoft hypervisor. So the hardware is actually virtualized. And this has all been done for us by University of the Algarve. And so we're able to provide virtual machines to the community, basically, and we can provide containerization and things like Docker and Singularity and Bioconda. We also have a setup of several nodes where we provide a high-performance queue that's actually running the wonderfully named Slurm, which is just a way of dividing jobs and resources amongst those jobs, equally, so that they can all run at the same time. We also do some project website hosting, which I'll talk about a bit later. But with respect to the high performance queue, I mean, we do a lot of genomics analysis and transcriptomics, metagenomics, et cetera, and we are able to actually do full genome assembly of prokaryotic and eukaryotic genomes. So this requires a lot of computing power, and one of the nodes that we have available for use is has a terabyte of RAM actually attached to it, so we can actually do these large eukaryotic assemblies. One that we've actually done within the last couple of weeks actually was Tetracelma striata, which is a unicellular alga. We had 197 gigabytes of packed biodata, and we used 56 CPUs for 7 days and 700 gigabytes of memory to analyze this data. And we got a very good assembly out of it, actually. It's actually very impressive. We've also been doing a lot of work on RNA-seq samples, and recently we've been analyzing 108 of these samples as well. So again, we have some pretty useful resources available to us in the community. In my particular domain, I do a lot of phylogenetic work, and often that involves doing long MCMC simulations, and these can run for 3 to 6 months. I think 6 months is the longest I've ever run one. But it shows that we have good uptime with these resources as well. We can actually run these things for a long time, so that's an advantage. We've done at least one project with ELIXIR on metagenomics. So we were involved with ELIXIR Accelerate program as part of Work Package 6. And the idea here was that we were going to actually build some synthetic data sets so that we could actually test the metagenomic pipelines that are being used to assemble these data sets. I mean, obviously, you need to have synthetic data sets so that you know how well these pipelines are doing actually with these assemblies. So our part of the project was to develop these data sets, and we have 6 of these semi-synthetic metagenomes that have different amounts of different organisms in there, where we know exactly what each read is and how they map to the actual final assembly. So we ran these things through 2 of the major metagenomic analysis pipelines, which is MGRAST and EMG. EMG is the EBI Metagenomics pipeline, which is now called EBI Magnify. And I think that's up to version 5 at the moment. So we ran these analyses, and we're still doing them now. We still intend to actually continue testing different pipelines. But at the time that we ran these analyses, both of these pipelines are doing a fairly good job at actually analyzing these data. So they were finding approximately 90% of all of the genera that we knew were in these samples, in these synthetic data sets, which is fairly good. As far as actually identifying, actually annotating the functional annotations of the synthetic data sets, they were both fairly poor, I would say. I mean, 50% accuracy is probably as good as you were going to get at the time, but I think overall, I mean, it's something that obviously can be built upon, and I think, as more and more metagenomes are actually published and the databases get bigger, I think that number will increase significantly. But that was at the time, which is about a year ago now. So that's one project we've been working on. The person who did most of the work there was Jean-Luc Demaro, who's my colleague at CCMA. Another bit of software that's actually been developed within the community is by Ricardo Let at IGC, and he's written this very useful bit of software that actually allows you to visualize the results of metagenomic analysis. And so I think this is really useful in the sense that you can actually visualize some of the characteristics of the assembly and the actual community composition. So if you're interested in actually using this, you can find links to this piece of software on the BioData.pt website. And also you can just search GitHub. I think it's on GitHub under the BioData.pt folders. The other thing I wanted to talk about was Ocean Sampling Day, which is a project that we've been involved with. In fact, it was Ricardo Costa, when he was at CCMAR and he's now at IST in Lisbon, who was mainly involved in this project. And Ocean Sampling Day is a worldwide initiative to sample seawater throughout, obviously throughout the planet, and to actually do some microbiome analyses of these samples. So this was being promoted at the European level by Micro3B, Simple Plus, and EMBRC again. It was started in 2014, and I think we actually had more sites in Portugal than any other European country. So we did really well in this, and we got a lot of data included in this project. Although I think there has been sampling in every year since. I think it's been very uneven. Some years we've had more data collected and some less. Originally, the idea was merely to, when you actually collecting these metagenomic samples, the idea was just to provide taxonomic inventories of these samples. And I think that was done originally within the first 2 or 3 years. But since 2019, these have been analyzed by EBI Magnify, and they're actually producing full metagenomic analysis, which is very impressive because they're very large data sets. So this was Ocean Sampling Day, and this is being forwarded again by EMBRC and Assemble Plus. And the idea here is that from 2020, and in fact the sampling has already begun on this, we're going to set up what are called genomic observatories, and these are basically a similar idea to Ocean Sampling Day, but these samples are taken more regularly. And so a genomic observatory may take a sample from the same place every month or even bi-weekly. And so these projects are going to be producing, these large data sets on a monthly or even bi-weekly basis. And so at the moment, one of the projects that we're trying to sort out with ELIXIR is to develop a pipeline that we can use through EMBRC to actually analyze these data before they're actually published. So, so we're working on that at the moment. The other thing that we also do is we host websites and different projects, and at least at CCMAR we have a lot of data from transcriptomics. We actually have hundreds of data sets, but at least for things like these 2 fishes that are example here, we have set up transcriptomic blast servers that we can find through CCMAR's website. And this was done by Bruno Laura at CCMAR. And our idea is that we eventually want, at least at CCMAR, to try and get all of our digital data online, and then actually have within the community links from BioData.pt to all of these different digital resources of all of the different marine institutes within Portugal, so that you can easily find these resources from a national site. So that's what we're working to there. Finally, I just want to point out this other really interesting site that we host here, and this is the Marine Forests website. And this is a worldwide community-based project where people actually provide data and inventories of different marine forests, macroalgae, fucus forests, seagrass meadows, etc. And I think this is a really interesting project, and it's been very worthwhile it actually being set up. And there are people from all around the world have contributed data to this project, which is really nice to see. And so one of those things this project actually does is look at seagrass meadows, which I guess brings me to a pretty awkward segue into the next part of our talk, which is going to be my colleague João, who's going to speak about a database of database of phenotypic data of seagrasses that we've been building as part of this community quite recently. So I'll hand over to João.
Can see.
Yes.
Okay. So good afternoon. Thank you for your presence. I'm going to talk about this project, the Seagrass Database. This project has been developing under a strict collaboration with the Marine Plant Ecology Research Group, namely Dr. Lisange and Dr. Carmen de Lisange. They are the ones that are helping us for this database to be useful for their research and for the research of people or researchers working on the same field. So basically the Seagrass database is a web resource to share datasets of seagrasses. We have already compiled information about 206 traits belonging to different groups, biochemistry, growth and development, morphology, and physiology. We have also already imported information about 86 species of seagrasses with some information about their taxonomy and some IDs for other databases like NCBI, Algabase, and FEL. So the website already have information about these species. We have developed the database following MEUP standards. MEUP stands for Minimum Information About the Plant Phenotype Experiment. It's an open project to harmonize the data, so the data becomes standard. One of the big advantage of this MEUP standard is that there is already a lot of tools to validate, to store and to disseminate MEUP compliant data. On the right, you have here a domain model for the MEUP database. They have 4 different domains that are fully required for the database to be MEUP compliant. There's the ones in orange are the ones that are required. Then the other ones, depending on your database, you can add or not. So in terms of technologies, we are developing the website using Django, which is a web framework for Python. We are also using jQuery for the data tables, for instance, some libraries of jQuery. We are going to deploy the website contained in a Docker. And we are also running a PostgreSQL Docker for the database of the website. In terms of roles and use cases, these are generic description of the use cases and the roles of the website. So the guest is allowed to register on the website. He is allowed also to search data and to download the data, only the public data, because the data will have 2 different states. One is to be public, other to be private. So only the owner will have access to that data. You can be an authorized user. You can also search data and download data, the public one and your own. You can authentify on and you can go to the dashboard. You can upload data, delete and update your own data, and you can edit the permissions on your own data. We have also another role, which is the administrator, that is basically for user management. So he's the one that approve or deny the requests for registrations on the website. We don't have a role, but maybe in the end we'll have an additional role, is the data curator. So when the data set becomes available to the public to have an intermediate step to check out if the data is correct. But right now we don't have that rule. So this is a simplified DB schema of the database, of the current state of the database. Some tables are not present and the fields are not shown, just because, for instance, for data value it will be impossible to show that entire data table, that table, sorry. So the central core of the application is the investigation table. Then the investigation table have a relation one-to-many to studies. So one investigation might have different studies, one or more studies, and the study will have a location, a taxonomy, and the trait values for that study. Another important thing is that the investigation will have an associated user, but the study might be the same user or a different user. So in this schema, you can have an investigation that is responsible of one user and different studies belonging to different users. So the landing page, on the landing page is shown a map with the location of the studies that are public and available for download. And depending on the zoom, you'll see different details. So for instance, on the left, you'll see that there are 4 studies already loaded for Portugal. But if you zoom in, then you'll see that 1 is in Aveiro and 3 are in Faro. So, and then we'll generate a pop-up to download that study. So once you require to have access to the dashboard of the website, you will receive an email for activation of your account, and then the administrator will approve or deny your request to have access to the dashboard. So this is a way also to control who can upload data. And the administrator can also disable or delete the user if required. The user also have an option to delete his own account and all the data associated with his account and his data. So to upload that, you actually first you need to look at to the template, download the template, and see the traits that are available for uploading. You can also filter if you don't need some of the traits and reduce a little bit the size of the template. The template will be just a CSV file with some headers, with some containing the traits that are possible to upload. And then you need to fill some fields on the metadata just to have more information about that data. And then you just fill your CSV file and upload to the website. After you upload the website, you can filter the data sets using some of the metadata fields. Right now, the investigation and the study are together, but in the end, we'll have 2 different layers for filtering. Even inside the application, we will only have access to your home data and the public data. So you cannot view others' data if they are not placed in public. When you open the details of a specific investigation, you can see all the studies that are associated with that investigation. You see a data table containing the traits and all the information of that investigation. You can download the CSV file, can download as an Excel. Right now, we don't have an option to download the metadata, but in the future, we need to include that in the next version. So this comes to the end, and what we need to do in the future is to have an option to download the CSV and the metadata associated as a text file in a zip package instead of downloading just a CSV file. Provide a public URL for the investigation study in text format and in JSON to be in a structured way, and to have also an option to download in a format compatible with ISO tools to be compliant with fair data. So.
Okay. Can I just say that this project grew out of a need that was brought to us by our colleagues Rui and Carmen, who've been working with colleagues throughout Europe to actually analyze this data. And normally these plant traits are placed within a database called TRI, which has been set up, I think, 20 years ago and has been expanded over the years. But this database doesn't really follow standards and ontologies, which is what we really wanted to do here. And I think that the TRI database didn't really accommodate the data for seagrasses the way that they wanted to use it. So this is why this project has become something we've taken on. So I think it could prove a useful tool to the seagrass community and seagrass ecologists. So that's what we're hoping with for this project.
Okay, thank you, Simon, and thank you, João. I'll seize the opportunity to ask the audience if there are any questions. If you want, you can put it on the chat. Maybe
Thank you, Simon and João, for this very nice talk. When you consider the Portuguese research community interested in marine resources, how do you see that this work can foster their research? Can you elaborate a bit on that?
Yeah, I think it's been very clear since BioData.pt has been, well, since I've been involved with it, that there are a lot of resources for marine data in Portugal, but they're all scattered. I mean, they're even scattered within my own institute. They're sitting on people's hard drives or, you know, in lab computers and such. And I think initially it's a matter of trying to get these resources actually published and available onto the web. And this has a lot to do with BioData.pt's drive for trying to make, you know, data accessible and fair, you know, fair data standards and open science. And I think that's a lot of what we can do is to actually promote open science and fair data access. And if more of this can happen, then I think, you know, trying to actually build a repository or a collation of all of the different marine digital data resources that are available would be very useful. But at the moment, I think it's very difficult to even find them. So, yeah.
Okay, I guess Carmen wants to add something.
Yes, hello. Can you hear me?
Yeah.
Yes, okay, thank you. Yes, well, I'm Carmen Santos. I work at SeaSmart, as you can see, and I'm involved in the project that João and Simon presented. And I think that this project can have a global impact in the way that we do research in the field of seagrass ecology. And this kind of data sets already exist for terrestrial plants. I think that Simon already mentioned that. And this is a gap of knowledge that we will cover. So I'm pretty sure that this will have an impact at the global scale for our field of study. And the traits, well, I don't know if the audience is familiar with this kind of table of data, but the traits are characteristics of the plants that allow us to understand all the processes, all the ecological processes. So we can have data on the leaf properties that can be used to understand, for example, the cycle of the nutrients at the ecosystem level, for example. So these are values at the plant level, but we can use it for studies at the ecosystem level. So I'm pretty sure that the Seagrass community will be very happy to see this project when it's out.
Yeah, I do have one question as well. So you said that you had 86 species on the SeaGrassDB, right?
Yes.
And can you tell me something about the geographical distribution of those species? Or do you not have that information?
I don't have, but maybe Carmen will be
Yes.
better because.
Yeah,
Sorry, Carmen.
The taxonomy of seagrasses is well, it's still in process, I would say. But yes, more or less we have 70s, 80s species, and they are distributed in the whole at the global scale. But most of them are concentrated in the tropical regions. Is the hotspot of seagrass biodiversity. Here, for example, in Europe, we have I don't know, maybe 9 species, counting with the Rupia species, which are not fully marine species. But for example, here in the Algarve, we have 4 or 5 species. I don't know how many Rupia species are. But it's not a high biodiversity if you compare with the rest of the planet, which has thousands of species.
Okay, thank you, Carmen. I think Ricardo has a question.
Okay, it's for Simon. Simon, it's a technical question about the direct cell news assembly. So you use PacBio. So you use PacBio, and you need all this coverage for PacBio, or you're combining with it?
No, it was massively over coverage. It was like times 400 or something.
Yeah, that's why I was asking. I thought it was maybe too much for that.
Yeah, we had. I think we had a whole column of PacBio. You could have done at least twice that. Yeah, no. So you're right. It was ridiculously over coverage.
Okay, I thought it was some problem like repeat regions on the genome, something like that, or too many plasmids.
No, we did get a very good deal with the actual sequencing company anyway, but still, you know, it was way more data than we needed.
Oh, okay, thanks.
But I should say that if anybody wants to actually use these infrastructure facilities to either get in contact with us directly or through the BioData.pt website, we're always willing to help, you know, with different analyses. We do install a lot of different software, and so it's not a problem.
Okay, thanks.
Okay, are there any more questions? Okay, so if not, I do have some announcements to make before we finish. Let me just share my screen. Okay, so for January 2021, we'll have our 4th BioData Talks. So we will have Kjell Petersen, which is the technical coordinator for ELIXIR Norway, and he is also from the University of Bergen. He will talk about the challenges to establish a federated EGA node and the experience of ELIXIR Norway itself on how they have established their local instance of EGA, which is the European Genome Archive, for those of you that don't know. So this will happen on the 21st of January from 2:30 p.m. to 4 p.m. Okay, so next announcement. On the 4th of February of 2021, we'll have our first Microbiome PT Summit in the scope of what Simon said, that the marine metagenomics community would make a transition to encompass all the microbiomes involved, and you are also free to register yourself, and we do at the event public. We can share it with you. And finally, we have opened yesterday the announcement for a new position at BioData.pt and ELIXIR Portugal for the seat for Chief Technical Officer or Technical Coordinator. And so if you are interested in somehow developing and coordinating our technical infrastructure and developing the technical strategy for implementing and developing the services of BioData and ELIXIR Portugal, please feel free to visit our site and apply for this position. So I would open the floor to questions. Maybe anyone has any doubt? Okay, so if not, I would like to thank everyone for being here with us, and especially Simon and João for this presentation. And see you next month. Thank you.
Bye, thank you.
Bye.
Bye, everyone.